Antibiotics are crucial for preventing the proliferation of bacteria that could be the cause of many serious infections. As you can see in Figure 1, there are many different mechanisms of antibiotics, which include inhibition of protein synthesis, inhibition of nucleic acid synthesis, inhibition of cell wall synthesis, disruption of cell membrane functions, and block pathways and inhibit metabolism.
Although antibiotics help fight infections, there is a growing problem of antibiotic resistance that is severely harming human health. The potency of certain antibiotics diminish due to overconsumption and mechanisms of antimicrobial resistance. The potency of certain antibiotics diminish due to overconsumption and mechanisms of antimicrobial resistance are well documented. Figure 2 displays the mechanisms of antibiotics when the bacteria they are targeting is resistant to them.
What exactly are rhamnolipids? Rhamnolipids are biosurfactants naturally produced by Pseudomonas aeruginosa. They promote the uptake and biodegradation of poorly soluble substrates, act as immune modulators and virulence factors, and have antimicrobial activities and are involved in surface motility and in bacterial biofilm development. They exhibit antimicrobial activities and surface active properties against several microbes, thereby showing promising applications in pharmaceuticals and therapeutics.
Rhamnolipids have also been used in many areas such as agriculture, therapeutics, and cosmetics. Figure 3 displays the biosynthesis process of rhamnolipids. On a genetic level, the biosynthesis of monorhamnolipids in Pseudomonas aeruginosa occurs through two sequential steps. First, the RHLA, which is encoded by the RHLA gene, is synthesized, and then the membrane-bound RHLB rhamnosyltransferase, which is encoded by the RHLB gene, uses molecules such as CTDPL-rhamnose and a HAA molecule as precursors, which yield monorhamnolipids.
Our experiment incorporates the findings from Lauren C. Rudolinsky at University of North Carolina. This study shows that rhamnolipids were shown to potentiate aminoglycoside activity against Staphylococcus aureus.
Let me start over. Figure 4 displays the results from this study and shows that rhamnolipids, when combined with the antibiotic tobramycin, significantly increase the antibiotic uptake in bacteria.
Altering membrane permeability to induce antibiotic uptake is an effective strategy against both gram-positive and gram-negative bacteria. Therefore, our objective is to identify a novel method of delivering antibiotics while reducing the possibility of antibiotic resistance. Our experimental hypothesis includes the synergistic effects of rhamnolipids and antibiotics will be greater when compared to rhamnolipids or antibiotics independently in susceptibility tests.
In our experiment, we used 6 different antibiotics. We used ampicillin, chloramphenicol, erythromycin, kanamycin, penicillin, and tetracycline. We elaborated on the mechanism of each antibiotics in our research paper. The gram-positive bacteria we used were Staphylococcus epidermidis, Bacillus megateria, Micrococcus luteus, and the gram-negative bacteria we used were E. coli MM294 strain. The possible infections caused by each bacteria are elaborated on in our research paper.
Our experiment was done over the course of 4 days. Our first step was to grow and culture the bacteria. Then, we diluted the rhamnolipid solution and mixed them with the antibiotics.
We then spread the bacteria from the liquid culture onto these plates to create a lawn. The antibiotic sensitivity tests were applied to agar as listed above.
This slide shows the pictorial analysis. The first column is plates with just bacteria. The second column, bacteria with rhamnolipids. The third column, bacteria with only antibiotics, erythromycin, chloramphenicol, and ampicillin. And the next column is just bacteria and antibiotics mixed with rhamnolipids. The second to last column shows the last 3 antibiotics, penicillin, tetracycline, and kanamycin. And the last column shows the last 3 antibiotics mixed with rhamnolipids against the specific bacteria.
The bacteria presented in each row is indicated by the electron microscopy picture to the left of the row. The zone of inhibition for most plates was greater when combined with antibiotics and rhamnolipids than when combined with rhamnolipids or antibiotics independently.
As seen in the graphs, antibiotics with rhamnolipids, shown in pink, have greater zone of inhibitions than just with antibiotics alone.
A blast analysis was also conducted, and it provided novel information regarding putative enzymes, homologous to RHLA and RHLB, found in Burkholderia pseudomallei. We hope to elucidate the role of putative proteins in Burkholderia pseudomallei through cloning and gene expression experiments. This picture shows the blast results for the RHLA gene, and these are the results for the RHLB gene.
The graph demonstrates kanamycin as having the greatest effect universally on the bacteria in the presence of rhamnolipid. This is because kanamycin is an aminoglycoside, which requires a PMF.
The study we used for reference from the University of North Carolina conducted their experiment with gram-positive bacteria and not gram-negative bacteria. E. coli was a gram-negative bacteria, and our experiment shows that rhamnolipids had permealizing effects on E. coli as well. Gram-negative bacteria have thinner peptidoglycan layers as their cell walls, but rhamnolipids had the ability to create pores in their cell walls as well. The results of our experiment ultimately support our experimental hypothesis. Rhamnolipids potentiated the tested antibiotics, and the zones of inhibition calculated for each bacteria support this claim. For E. coli, the rhamnolipids increase the zone of inhibition for all antibiotics except penicillin. For Bacillus megaterium, the rhamnolipids increase the zone of inhibition for antibiotics except for erythromycin and penicillin. The rhamnolipids were the most effective against Staphylococcus epidermidis. Staphylococcus luteus, the effects of rhamnolipids against this bacteria with ampicillin, chloramphenicol, and erythromycin were undeterminable because the components of the plate were jumbled in the incubator. However, the rhamnolipids were still effective and did increase the zone of inhibition for kanamycin, penicillin, and tetracycline.
The rhamnolipid combination represents a new approach for resolving chronic or relapsing infection and slowing the spread of resistance. The blast results demonstrated on the past slides determined a strong match between Pseudomonas, RHLA, and RHLB genes, and putative proteins expressed by Burkholderia.
Not only can you apply rhamnolipids to antibiotics, there are many different uses of rhamnolipids. Our future research includes the use of different antibiotics and different strains of bacteria, different types of rhamnolipid concentrations, using rhamnolipids as a potential biocontrol agent for crop culture protection. This future research will demonstrate the interaction of natural rhamnolipids with the plant and fungi membrane models at the molecular
Because
scale.
the rhamnolipids showed permeabilizing effects against the bacteria with antibiotics, it is likely that they will also show these effects against fungi membranes and can be used to replace biopesticides that can be toxic. The results from our blast analysis show that the RHLA and RHLB genes from Pseudomonas aeruginosa were homologous to the Burkholderia pathogenetic strain. Our future applications can include cloning these putative genes from Burkholderia into E. coli strain to express the genes in E. coli and test for expression and synthesis of rhamnolipids, create an additional manufacturer of rhamnolipids.
Rhamnolipids can also be used to combat cancer. Monorhamnolipids